The SPIDER program is it's the serial patient-derived xenografts to understand cancer therapy resistance program. So really what we're trying to do is trying to understand both the efficacy of new drugs that are targeted agents and even immunotherapy agents, and what resistance develops. So how can we overcome that resistance? And we want to learn these things as quickly as possible. So the SPIDER program involves patient or research-based patient tumor biopsies. So we do a biopsy prior to the start of a new treatment. And the patients who go on this trial are going on with a particular target that we're interested in targeting and a particular drug. So there's a pair, a drug target pair. Do a biopsy before of this and implant these tumor tissue into immunodeficient mice. And so we call those patient-derived xenografts. When we establish those patient-derived xenograft models, we're able to treat the model in the same way that we treated the patient. So we have the molecular alteration in the drug pair. And we're able to look at that and say what type of resistance develops. The advantage of patient-derived xenografts compared to cell line models is they recapitulate a lot more of the diversity of cells that go into a tumor. We know that tumors are not monomorphic populations. They have stroma and many other things that are involved. And so we're able to sort of recapitulate that. And so we think it's very likely that these models will better recapitulate what actually happens in the human. And then when the patient becomes resistant to their treatment, so they've become resistant because of the acquiring some sort of resistance mechanism, we do a biopsy again at that time and establish a model that is sort of the resistance model in the same way that we did at the beginning. So we want to ask the questions, does the resistance mechanism that develops when we treat the human patient the same as the resistance mechanisms that we develop when we treat the xenograft in the PDX model?